Hello everyone, welcome to Dr. Vidya's Science Videos. In today's video, I'll be talking about how coronavirus enters our cells. This microscopic virus enters our body through droplets. As you all know, our body is made up of trillions of cells. These cells are basically functional units of our body. So, the viruses which have entered our body cannot replicate by itself. So, now this virus infects the host cell, uses different mechanisms or machineries of the host cell to replicate and increase its infectivity. Now the question is, how does it enter the host cell? This coronavirus has a unique structure which helps its entry into the cell. These virus particles are nothing but small collection of genetic code. It is either DNA or RNA. As you are seeing in the picture, this coronavirus is spherical in shape, which is coated by three types of proteins. The first one is envelope protein, the second one is membrane protein, and the third one is spike protein. This spike protein again has two subunits, S1 and S2. S1 unit, spike 1 subunit, contains receptor binding domain, which is responsible for initial attachment of this virus to the surface of the host cell. As I already told you, our body is made up of trillions of cells. On most of this cell surface, there are receptors called as ACE2 receptors or angiotensin converting enzyme receptors. Now, this S1 subunit of spike protein has receptor binding domain which binds to this angiotensin converting enzyme receptors. And the second subunit of spike protein, that is S2, is responsible for membrane fusion and entry into the cell. Now, the virus has made its way into the cell. But this virus has a problem. It cannot replicate by itself to cause or increase the infectivity. So what does it do? It misuses two important cellular mechanisms or machineries. Those two mechanisms are glycolysis and glycosylation. Glycolysis is a process by which our cells produce energy in the form of ATP by breaking down glucose. The main source of glucose, as we all know, is through diet and gluconeogenesis. Gluconeogenesis is a process by which glucose is produced by our body through non-carbohydrate substrates. Now these viruses, after invading our cells, increases the uptake of glucose by infected cells. And in turn, it upregulates glycolysis. So, upregulation of glycolysis leads to more energy production, which these viruses use for its replication. And another cellular mechanism that viruses misuse is glycosylation. Glycosylation is a process by which our cells produce glycoproteins. These glycoproteins are essential and play a major role in structure and functioning of our body. Viruses in infected cells causes upregulation of glycosylation, which leads to formation of viral glycoproteins, which are necessary for replication and to increase the infectivity of the virus. While doing so, it often kills the host cell or causes damage. Now we have understood how this virus enters our cell and disturbs the cellular mechanism. In the next video, we shall see how exactly it upregulates glycolysis and glycosylation.